Hello everyone, I'm Dr. Aravindh, Department of Pharmacology. Here we are going to discuss about some topics regarding general pharmacology. So these are the topics we are going to discuss now. Therapeutic drug monitoring, first one. Next, therapeutic window, drug development, essential drugs concept, rational use of drugs. So first we will discuss about therapeutic drug monitoring. So here patients, one patient is coming, for example, diabetic patient, for him we are giving anti-diabetic drugs. So after giving drugs, we will check the blood glucose level. So if the patient are taking tablet anti-diabetic drugs regularly, uh, his blood glucose level will confirm go to normal level, or from it will reduce from the higher level. So in this between the time, those medications, they will enter the patient's blood circulation and they will do their work. So in the time we have to check the drug or drug metabolite in the patient's blood. If it is within the normal range, they will produce the expected response, like decreasing blood glucose level. If it is more than the normal range, it will produce some side effect toxicity. If it is less than the normal range, it will produce some uh, like it won't produce efficient response. So first we will define what is TDM. So here clinical laboratory measurement of specific drugs at specific time intervals to maintain constant concentration of the drug in patient's blood to optimize the individual dosage regimen. So what we are here going to do, we will give some drug. So in the drug, we will check the measurement of the drug level in the patient's blood at a specific time interval. We will check. So depending upon that one, we will give the drug in a correct dose. Next, what is the aim of TDM? So because after checking, we will give the dosage of the drug perfectly. So because of that one, patient will develop a good response. So to optimize therapeutic responses, like preventing seizure. For example, if you are giving anti-seizure drug, it will produce, it will prevent the seizure, arrhythmias like that. And the second aim of the TDM, so we are monitoring the drug level in the blood regularly. So because of that one, we can avoid drug related toxicities like nephrotoxicity by aminoglycosides like amikacin, gentamicin, erythromycin, azithromycin. Like those are the drugs. If you give, the patient may develop nephrotoxicity. By correctly monitoring the drug level in the blood, we can avoid these kind of toxicities. For these two main purposes, we are doing therapeutic drug monitoring. Next, therapeutic index or therapeutic ratio. This is the one we have to know, very important. So therapeutic index is equal to LD50 by ED50. LD means lethal dose, ED means effective dose. So LD50 means one acute dose that is lethal to 50% of the animals. Lethal means a danger. It will produce more side effect like that. So if you take 100 animals, in that dose, it will produce 50% of the animals, they will become sick or they will die. In ED50 means acute dose that is effective to 50% of the animals. So 50% of the animals will get improved from the disease. This is ED50 and LD50. So for the safer drug, that therapeutic index, it should be more than 1. So more than 1 means like if you are taking paracetamol effective dose 500 mg we are using. Lethal dose 4 grams. So 4 grams by 0.5 is equal to 8. It is more than 1. So more than 1, paracetamol is a safer drug. So like that, we have to calculate the therapeutic index and we have to maintain the therapeutic ratio. So next one, therapeutic window. Here we are giving same dosage regimen to patients. Like one, we will take the patients, same dosage regimen. We will give some patients. In those patients, there will be a high plasma concentration of the drug and it will produce toxicity because it is more high. At the same dosage drug, we are giving another group of patients, those patients also they will develop low plasma concentration of the drug, and it will lead to insufficient response. Here, same drug we are giving, but the response are varying because of their most of the time it will be genetics. Next, we have to maintain this therapeutic window. So look here. So here, where less dose, there will be suboptimal. That means there is no response, or the response will be insufficient. For example, hypertensive patient, we are giving antihypertensive drugs, but its dosage is not enough. So the BP is not controlled. So here, if we are giving more dose, it will produce some toxic effect. So we have to maintain the drug level in our blood in a normal range. Within that normal range, it will produce correct response. Like it will decrease blood pressure like that. So we have to know about the therapeutic window also. So next, these are the some examples. So theophylline we are giving for asthmatic patients. So here that range it should be 5 to 10 microgram per ml. So we should maintain that level. If it is more than 10, it will produce side effect, more side effect. If it is less than 5, it won't produce any effect. Like for carbamazepine, 4 to 10 microgram per ml. For phenytoin, 10 to 20 microgram per ml. For lithium, 0.8 to 1.4 milliequivalents per ml. So within this is some of the examples. We have to maintain within this normal range. Next, this TDM is needed when. So whenever, what are the times we need this TDM? First one, that drugs with a narrow therapeutic range. So narrow therapeutic range means they are somewhat dangerous drugs. So we have to maintain the blood levels of drugs. So regularly we have to check. Next one, the direct relationship between the drug or metabolite levels in plasma and effect. Like we are giving antihypertensive drugs. For that, the blood pressure will be reduced. So there is a relationship. At the same time, for diabetic patients, we are giving diabetic drug. At the same time, the effect, there will be a reduced blood glucose level. So there is a relationship. In these conditions, we can do TDM. Then the therapeutic effect cannot be readily assessed by quantitative measures or clinical observations. So here we can correlate like that. Also we can do. At the same time, if the like for anticoagulants, we are giving heparin for one patient. For him, we can do PTINR. Like that, we can do quantitative. Quantitative means we can measure. So like doing a blood investigations, we can assess the effect of the drug. Like a heparin, like that we are giving, we can quantify. Our clinical observations, like previously told, antihypertensive drugs we are giving, the BP and that the blood pressure it will become reduced. So like that, those effect it cannot be assessed. In those conditions, we will use this TDM. So here the relation should be there. Next one, that effect it cannot be assessed easily. So those drugs we will do TDM. Next, analytical text. If you are giving one drug, we should know the method to measure the drug level in the blood. For that method, it should be available. If it is not available, we can't do TDM. If it is available, we can do TDM. Next, when the TDM is not needed, if it is the drug with a wide therapeutic range. Wide therapeutic range means it's a safer drug. So there is no need to monitor. Next, unrelated. So the clinical effect, if it is unrelated to the concentration, like hit and run drugs. What is hit and run drugs? Like pantoprazole we are giving for ulcer. But if we give within one day, it will come, it will act and it will go. But its effect will stay longer for 1 week or 2 weeks, like that. So here that the clinical outcome is unrelated to the concentration of the blood, drug in the blood. So there is no relation, but the effect will be there. So this kind of time we don't need a TDM, and the effects can be clinically quantified. So those are effects we are quantifying. Quantifying means previously I told for hypertensive patient we are giving antihypertensive drugs. So because of that one, the blood pressure is reduced. So here we can clinically we can see, we can observe the blood pressure is decreasing. So in these kind of conditions, we don't need a therapeutic drug monitoring. Next, types of sample and timing of sample collection. Okay, what are the types of sample we can collect? So major we are taking serum or plasma. Next, urine also we are taking for benzodiazepines like a diazepam, midazolam, lorazepam, those kind of drugs. Sweat, cocaine, heroin. Then breathe for alcohol. Next, saliva. Saliva for lithium, carbamazepine, amitriptyline, phenytoin. For these kind of drugs, we can take saliva and we can do analytical techniques, methods to check how much lithium concentration or how much phenytoin concentration. Like that, we can check. Next, timing of a sample collection. So there will be a timing for short acting drugs. For those drugs, we have to take the sample immediately before the next oral dose. So like afternoon, the patient going to take the dose. Before that, we have to take the sample immediately before the next oral dose. For longer acting drugs, we have to take after 5 t of time of interval, like a pay for longer acting drug patient is taking. So after 5 t of, for every drug there will be a t of. So we have to calculate a 5 t of time. If for example one drug t of some 5 hours, so 5 t of means 5 cross 5 25. So after 25 hours we have to take the sample for longer acting drugs. So this is timing of sample collection. Next, these are the drugs which needed TDM, like a drug monitoring. So amiodarone, phenytoin, procainamide, propranolol, gentamicin, amikacin, lithium, digoxin, valproate, carbamazepine, theophylline, methotrexate, cyclosporine. These are some of the drugs which need therapeutic drug monitoring because they are narrow therapeutic drugs. At the same time, we have to see, we have to monitor the therapeutic range. Next, analytical methods. So we took sample and how we are going to analyze means there are some of the techniques we are using in biochemistry. That one, thin layer chromatography, high performance liquid chromatography, that is HPLC, with the gas liquid chromatography. This is a standard one we are using. Next, radio immunoassay, enzyme linked immunosorbent assay, that is ELISA. So these are the some of the methods we are using to measure the drug or its metabolite level in the blood or some other samples. Okay. Next topic, drug development. Drug development means for some particular disease, we are developing new drugs. So how the drugs are developing in industries, we are going to see about one by one. So here, development of the new drug from its genesis in the chemical laboratory. So first in the laboratory, we will develop. From there, its final acceptance as a safe and useful therapeutic agent under Schedule Y of Drugs and Cosmetic Act 2001. So depending on this act, we are producing new drug from the lab. From that one to we are using as a tablet. So these steps involved in the drug development. In that development, we have three main phases. First one, drug discovery phase. Second one, preclinical phase. Third one, clinical phase. So drug discovery phase, we have to identify the drug molecule. So okay, which it may be a natural from plant we can get, or it may be synthetic. We will create by chemicals. So this is a drug discovery phase. After that one, we will check it with the animals. So we will do animal studies in preclinical phase. Next, after animal studies, we will do those studies with the humans. That is clinical phase. In these three phases, after that, the new drug will come to market for the disease, for a particular disease. So here, this is a picture. So first, drug discovery. Drug discovery, obviously, it will take around 2 to 5 years. Then preclinical phase, it will take 1.5 to 2 years. I will explain this one later. So drug discovery phase. So serendipity. Serendipity means it's a lucky one. Lucky one means already we are using some old drugs. It's that the old drugs side effect or it will be used for some other use. For example, lignocaine. Lignocaine we are using as a local anesthetic purpose. But the same lignocaine it can be used for arrhythmias also. So it is a older drug as a lignocaine, but it is used for anesthesia. But a new user we found out what it is used for arrhythmia. So that is called a serendipity. It's a lucky finding. So only this kind of thing we are identifying new drugs. Next one, target-centered approach. Target-centered approach means we are a target, like we know what is the target. So based on the target, we are synthesizing the drugs. For example, AC inhibitors, enalapril, captopril, lisinopril, those are the AC inhibitors. So here are used for hypertension. So in hypertension, this ACE enzyme, angiotensin converting enzyme, this one plays an important role in hypertension. So what we will do, so this angiotensin converting enzyme is a culprit. So this is the target. What we are going to do, we will inhibit this enzyme. So this is a target. So to inhibit this one, we are creating the drugs. So this is called the target-centered approach, AC inhibitors. Next, a compound-centered approach. Compound-centered approach means like already one drug will be there. For hypertension, we are using propranolol, beta blocker. So same propranolol, we will use the same methods with some change. We will use it for better benefit. Baby cost will be low. The efficacy, its effect will be more. So like that, we can create drugs. Those are all called me too drugs because propranolol is a prototype drug. Based on that one, we are creating so many drugs. This is called a compound centered approach. So after that, lead optimization. Lead optimization means, so we are creating some lead particles. That means a drug. These drugs, it will be used in animal studies. So drug discovery phase, obviously it will take 2 to 5 years. Next, a preclinical phase. In that preclinical phase, those drug molecule, it will be used in animal studies. So we will give it to animals and we will do studies. So it will take 1.5 to 2 years to get the data regarding the pharmacokinetics, pharmacodynamics, toxicology, like how the drug going to animal body, how it get eliminated, how it is absorbed, then the drugs, what it will do to the animal, those things. So because of the drug, what are the side effect, adverse effect happening in that animal? So these are all the studies we will do in animals and we will note the data. Okay, if it is okay in all tests, it's getting pass. That means that drug will be used for human trials. So promising compound for human trials. Like from drug discovery phase, we will identify some 3 or 4 molecules. We will do test with the animals. In that, maybe 1 or 2 drug molecule will become successfully to human trials. So next, third phase, clinical trial phase. So here the drug molecule tested in the human, like a patients. So for that, we need to get the permission from the Drug Controller General, Government of India. We have to get the permission from this authority. So to get approval, it will take around 1.5 years. So after that, we will start clinical trial phase. In that clinical trial, it will take mostly 5 to 7 years. So in phase 1, there will be non-blind open trials. So that means what is the drug? All workers will know. So here 25 to 100 healthy volunteers we are taking. We will get the consent from the health volunteers. With those healthy volunteers, we will give the drug. What we will do, because the patient is healthy, so we can see the pharmacokinetic and the pharmacodynamic aspect of the drug molecule. At the same time, we can assess the safety, tolerability, and a safer clinical dose for human volunteers. Like that, we can check, okay, how much dose we can give for the humans. Like that, we can check how much dose pay that human can tolerate. Those things we can identify, we can assess. Next, phase 2. In that phase 2, we are 200 to 400 patients here we are taking. Well, like if I'm identifying, I'm discovering on a drug for hypertensive patient. So here I will gather 200 to 400 patients of hypertension. So with those people, I will give the new drug molecule and I will see the response, how much safety, how much efficacy. Efficacy means it should produce the response of the new drug in the specific disease. So I will see how much the response is coming. Next, third phase. If it is clearing that 200 to 400 people, then I will do the same study with a large scale, like 1,000 to 5,000 plus patients. I will do with double-blinded trials. What is double-blinded trials? Here, the patient don't know what the drug they are getting. At the same time, the doctor also don't know what the new drug he is prescribing. This is called a double-blinded trials. So multi-centered, like not in a one center, some 5, 6 centers, they will gather these much patients and they will give drugs and they will check how the drugs are working. Okay, if it is working nicely, then we have to do NDA, new drug application. So after I'm identifying one drug, I'm doing these many trials and it get working. So I have to give this a new drug application form to the licensing authority. They have to give license to market the drug in the country. So after that, the drug get approval and it will be marketed in the country. So after that one, we have to do post-marketing surveillance. What is post-marketing surveillance? After giving to the patients or the public, how much response the patients are getting. Next, what are the side effects they are getting? Maybe at the trial time, nobody didn't get any side effect. But after enter into the market, maybe some patients may develop some side effect. So we have to note all the side effect and we have to search for the resolution. Okay, what other things we can do to reduce the side effect. So based on that drug, I will prepare another one drug which will not produce the same side effect, like that. Next, I have to do submission of PSUR, periodic safety update report, like regularly by the sponsor for 4 years. For next 4 years, that industrial sponsor, they have to submit a PSUR, that report, what are the side effects happened, how much people get improved, like that, that report I have to submit. Next topic, we are going to see about concept of essential medicine. Essential means need, necessary. I need these kind of drugs. So what are the concept? So as per WHO, World Health Organization, so these are the drugs, they should satisfy healthcare needs of majority of population. Okay, so in a community, there will be some diseases. For some disease, we will use some drugs only. With some drugs, the disease will be cleared. In the majority of the people will come like ulcer or vomiting, like that they will come. For that we will give tablet. So with the tablet, the patient will become clear. So it should satisfy the healthcare needs of majority of population and it should be available at all times. So like pantoprazole, ranitidine, like that we are taking. It should be available at all time in adequate amount and in appropriate dosage form. Like it should be in tablet or it should be in syrup. Like that it should be there with assured quality, because the quality is more important. It should be with good quality and at affordable price. So it should be affordable to the all kind of people. So here only a handful of drugs are required to treat most of the diseases. So we have so many drugs, thousands of drugs we are having, but we are not using all those drugs. Mainly we are using some 300 or 400 drugs only we are using. So we are 400 drugs we are using in most of the people for most of the time. So for that, WHO, it published one list, essential medicines list in 2017, SAT 20th model. So like that, every country, they can prepare their own list. It may differ from other countries because in India, there is more snakes. So because of we are using anti-snake venom vaccine, but in New Zealand, there are no snakes. So for them, it might not necessary. But for India, like our country, we need anti-snake venom. So this list will be varying from country to country. So in India also, we developed a national list of essential medicine. It is released by the Directorate General of Health Services. Latest released in 2015. So in that list, we are having 376 drugs. Those are all essential medicines. So every hospital should possess, should have the drugs. Next, rational use of drugs. What is a rational use of drugs? Means we have to use the drugs properly, correctly. So we are based on the rule of right. So right drug to the right patient in right dosage form at the right cost with the right documentation for right time. So here patient is coming and complaining. So we have to diagnose the patient. So based on the diagnosis, we have to give the right drug to the right patient. If it is a diabetic patient, we have to give diabetic drug in a right dosage form. Then only the diabetes will get reduced. So right dosage form, right cost. It should be affordable because the patient may be poor. So right drug we should give with the right cost, which should be documented rightly at for right time. For example, if patient taking antibiotic, he should take 5 days, but the patient may take only 2 3 days. Within that time, patient may get improved, so he will stop the drug. It should not happen like this. So we have to tell the patient like take the drug perfectly 5 days. So like that it should be on right. If anything occur wrong means it is called the irrational use. So we have to confirm this one. Next, it should also fulfill the same criteria. What is same criteria? The drug should be safety, affordability, like affordable at a lower cost. Then it should be needed for the patient, and it should be a efficacy of the drug. If the drug fulfilling this criteria, it should be used in a right way. So this is rational use of drugs. Okay, thank you. So if you have any doubt, you can comment. Thank you.